Thank you for joining Once Changing the World. I'm your host, Eddie Evil, and today I'm delighted and honored to have with me Dr. Brett Kagan, whose expertise lies in stem cell research, regenerative medicine, and neurotechnology. And he's currently the Chief Technology Officer of Cortical Labs, the team pioneering one of the world's first commercial biocomputer. A computer that's not just silicon circuits, but alive, grown from human neurons, learning like a brain, and potentially might one day play a role in revolutionizing the approach towards building general purpose machines. So, Brett, really appreciate you taking time and being part of the podcast. So, before we kind of get into biocomputers, wanted to get your thoughts on the computer itself, because it's taken like possibly 100 years for the computers to evolve, with code using just zeros and ones. So it'll be great if you could explain my audience a computer, a classical computer, how do they work, and what do you think about these current LLM architectures, with the scaling hypothesis? Do you think that's what might get us to AGI?
My goodness, a lot of big questions there. Where to begin? I think I might very broadly not dive too much into how a classical computer works in detail, but say at a very high level that key aspect of them, of course, is the basis of a silicon transistor, which ultimately operates on the principles of binary 0 and 1. And through that, you can build up a whole set of logic circuits that can exist. And these can be assembled into very interesting architectures, the most common of which one is broadly referred to as a von Neumann architecture, which involves basically shuffling around processing and memory very quickly. This can be incredibly powerful, as we've seen. There's no doubt that anyone could deny the impact that these architectures have had in revolutionizing our world, but they are energy intensive and relatively, they can have some limitations for particular things. Now, I think perhaps at the pinnacle of what's been achieved in many ways, although not all, are the current Gen AI approaches, as people like to call them, such as LLMs, diffusion models, broadly speaking. Now, you mentioned the scaling factor of that. And obviously, one of the big things that's taken LLMs from a sort of janky chatbot that was sort of passable as something to spend your time on, but not anything as capable as what we've seen now, is the absolutely mammoth scaling that's gone on. The question is, how extensible is scaling? And what I think we're seeing, right, and that's an open question, and it's a fair question, and if you're prepared to invest many billions of dollars, you can answer that question, and people have been prepared to invest many billions of dollars, and I think we're starting to get an answer, and I think what we're seeing now is that, indeed, we are hitting a wall. That's what scaling can achieve, and this was inevitable, in my opinion. It was predicted by many experts. It was not talked about so much by the ones with the media attention or raising the money for perhaps obvious reasons. But the question is, now that we've seen what scale can get us, and perhaps also the limits of what scale can get us, what's the next step? And that's sort of where we fit into the picture, saying the architecture that has led to things like ChatGPT or Claude, so forth, all these architectures, is there another approach, maybe another substrate, another information processing substrate will actually get us from incredibly helpful but ultimately limited AI, if you want to use that term, to something maybe that is more general? And for us, that's why we became interested long before LLMs were part of the scene, but it's why we were always interested in biology, because biology is this ground truth of a general intelligence. Whether you're talking about a worm, a bee, a dog, or even a human, that we all show a level of general intelligence, and how do we do it with biology? So maybe that's a way to sort of fit some of those very big questions you asked into a bit of a narrative.
Right. Yeah. So interesting. Yes. I think, I mean, these computers, these binaries, which use zeros and one, these classical computers, I mean, it's completely upended a world. And with these large language models, currently with the scaling hypothesis, many, I mean, I think the Anthropics, the OpenAIs, the DeepMinds, the Meta, not Meta, the others, they actually thought that, the scaling would stand true. You throw in a lot of data and eventually, I mean, you would get possibly general purpose machines, but yes, I think we be seeing a limit over there. But even with these these scaling hypotheses, with this just these statistical models where these you throw in a lot of data and you get a lot of data. I mean, the world has completely changed. I mean, who would have thought? I mean, it's just possibly 3 years, 2022 November, that's when ChatGPT 3 kind of launched, and it's 2025. In this short span of time, I think the most groundbreaking technological innovation that we have seen has got to be these large language models. Yes, there have been these other group of people who've been saying from day one that possibly these data and statistical models might not be the right approach, and maybe we might need to kind of reverse engineer the brain of sorts, because uh, 20 watts, that's what our brain uses, and it's doing surprising things, and then why aren't we doing that? So yes, there are a lot of people around the world who are kind of looking at that approach. Want to kind of get into that before we kind of get into maybe like reverse engineering the brain or the approach that you're talking about. Wanted to ground my audience in what you're doing, because in classical computers we use zeros and one binaries, but with here we are with what you're doing at Cortical Labs is using brain organoids, which are basically mini brains which are cultured on these micro electrode arrays. Will be great if you can elaborate on what are these brain organoids, how do you culture these cells into organoids, and how are these organoids finally leveraged for computation?
Yeah. Okay. Yeah. So I think, like, I'll comment just on the very first part you said uh, about LLMs and so forth being quite astonishing and or at least very capable. And absolutely, I agree with you. But I think it's the right tool for the right job. And so if the focus was we're going to build a tool that's going to help do really, really good documentation and can provide answers and be good at synthesizing information, that's useful. It's a very specific type of intelligence that as a tool is incredibly useful. And I don't think anyone should begrudge someone who built those tools. I think where the issue and the pushback came from is the idea that this was going to lead to general intelligence or super intelligence, right? And I think it's really good to be aware that criticizing that particular claim is not the same as saying these tools aren't useful or anything like that. They are useful. I use them myself. They're brilliant to do a whole range of things that would otherwise have just been busy work that anyone could have done, right? There's lots of things that fall into that. Job advertisements, doing up Excel spreadsheets. There's a whole range of things. So it's the right tool for the right job. And I think the only thing comes down to what is it you're really building. Now, we fall into that category as well. And I think it's good there to mention brain organoids, which are simplified 3D models of the brain, but they're not the same as many brains, right? Again, being aware of what we're actually doing is important. They're models of the brain. And actually, a really interesting point here is that that's just one approach. So we focus on, yes, we use organoids, and this is a push that's recently we've been part of, but been led out of Johns Hopkins University. And this has been called organoid intelligence. And it's the idea of using these representations of a brain as a way to process information. Now, these three-dimensional cell cultures are basically formed through a combination of different neural or brain cells that we can grow. The way we grow them specifically is from something called an induced pluripotent stem cell. So we take someone's blood or skin, in our case we use blood, same as what you'd give to a doctor for any routine test. We isolate some cells from that blood, we turn them into stem cells, and then from there we can go forward into any type of cell that you might want. Of course, not every type of cell people have optimized the best process. This is part of what we do, but there's a large range of neural brain cells that we can grow. So we grow cortical cells, we grow a range of other cells that we're interested in. And from that, we can choose, right? Here we have a material, much like the silicon transistor as a material, and you can assemble that in a range of different ways. The neural cell tissue at its precursor level is a material. And so one approach is to say, well, brains work well. They work in bees and they work in humans. Maybe we don't want a human brain. Maybe we're going to try and build up to bee-level complexity. Who knows? Let's build something like a brain and pump information into that. You can do that through a range of methods, chemical, electrical. We choose to use mainly electrical as a shared language between the silicon and the biology. That's one approach. The other approach you can say is, all right, if we have this material, what if we built something that wasn't at all like a brain, but still had really unique properties? And this is called bioengineered intelligence. And we've been really pushing down this avenue as well, because it's highly controllable and highly reproducible. And with this, what the goal is to create circuits with particular properties, much as you might want in a computer, but with the innate computational capabilities of neurons or neural systems, which far exceeds that of a silicon transistor. So you may end up with something that's far more powerful with less energy requirement. Not quite a brain, not quite a computer, but something engineered to do intelligence and information processing. So there's multiple options.
Right. Interesting. So here you were saying that, you using multiple approaches. One is organoids, and then there's bioengineered intelligence, which are real circuits, Will be great if you can elaborate on this. And Brett, it will be great if you could also go back to 2021, where Cortical Labs at first used these mini lab-grown brains which could play Pongs. How did you manage to do that? When you say that, these mini clumps of brains could play Pong, how could they sense these games or these Pong balls? And how challenging was the process of making these mini brains learn how to play Pong?
It was fairly challenging. We were a very small team back there. We raised a seed round in 2023, which is when we really started growing the company. There was just a very few of us back then. Before that, so in short, what we looked at doing is asking the question initially: can brain cells in a dish learn to do anything at all? Because it wasn't clear that they could. It's all well and good that we can look at, as I said, bees, cats, humans, and say they all use brains, amazing. But actually, so we knew it was possible, but how remained a really big question. How do you get brain cells to do something? So one of the very early ideas that have been around for a really long time, was popularized in the movie The Matrix, right? The idea that a lot of brain communication is electrical signals being propagated throughout the network. And so we wanted to use technology called microelectrode arrays, or MEA, as a way to actually communicate with the cells. And we hypothesized that if we structured our information input, right, our small electrical pulses, and we stimulated them, we could convey information to them. And if we read their electrical activity, which we could also do, we might be able to decode that as some sort of action. And so we were inspired by a number of processes that go on in the existing brain, and we encoded information in a way that initially started out inspired by the way that our visual cortex works, which is called topographic mapping, which is very simple. Basically, there's a one-to-one representation in our brains at some level about, say, you see something on the left, some cells on the left, well, there's a whole thing there, but on the left of the visual map, in any case, will light up in the brain, and there's a one-to-one mapping with the brain and the eye. And so this is called topographic mapping, and it goes through a whole bunch of layers. Of course, there's a lot of complexity in there, but that's a very simple approach. We ended up adding in a frequency domain, so in fact, it better came to represent the way that, say, a rodent's whiskers work. A rodent's whisker has a topographic map, but it also, so a spatial map, but it also has a temporal or a rate-based element. So the more pressure, the faster the neurons fire. So this is what we did. We basically said, all right, if the ball is on the left of the paddle, we're interested in this game Pong. As a way to solve it, we will stimulate on the left side of the region, and the closer it gets to the paddle or the wall, the quicker we'll stimulate. So it kind of represented some aspects in biology. And then we sort of said, alrighty, it misses or it hits the ball. These are the two options. How do you actually say that we want it to hit the ball? Because even if the cells could sense the ball relative to the paddle, why should they want to hit it, right? And so we looked at a number of different theories, and we came to really like some of the ideas of something called the free energy principle. And this basically said at a very high level, because I know we've only got so much time today, neural systems are going to try to minimize their what's called information entropy, or in a simpler term, they want to be able to predict their environment. Now, there's two ways to predict an environment. One is you get better at predicting the environment no matter what you do. Or two, you change your actions to try to control the environment to make it more predictable. There's not really any other option there. And so we said, what if we take away one of these options? What if we said that every time the ball is missed, you will receive random stimulation, something that can't be predicted? Under the theory, the cells then should change their activity patterns if the activity patterns is connected to the way the paddle moved to be able to hit the ball more. Because if they don't, they're getting random information they can't predict. Now, we were very agnostic. We weren't setting out to prove this theory, simply to test it. We worked with leaders in the field like Professor Carl Friston and Adil Razi over at Monash, who have worked on this theory for a long time, and we agreed that this was a good test of the theory, and if we found data to support the theory, that's one thing, and if we found data to challenge the theory, that's the other. So we're doing good science. In the end, we found data to support the theory, suggesting that this indeed is an aspect that drives neural activity. So that's basically how we, at a very, very, very high level, looked at setting it up. But I should say back then, we weren't using organoids, and we weren't using these bioengineered circuits. We were using these simple flat layer of cells called a monolayer. So it was actually an even simpler circuit than anything we've talked about so far. So we were very early back then. Everything we did was quite simple, and really the question was, can you get cells to do anything at all? Let's start with Pong and see if we can get any evidence that they can get better at Pong over time. And we were quite relieved to find quite strong evidence that these cells were incredibly adaptable and responsive over time.
What's the progress been over the years? Because you mentioned that back then in 2021, you were using flat two-dimensional cells, and right now these brain organoids, cultured, bioengineered intelligence, these are three-dimensional organoid cells. And what's the progress been? From training it to play Pong. What can it do at this point in time? And maybe it'll be great if you could also elaborate on these, these brain organoids, because I think that itself, that space is growing extremely rapidly. What's the volume of the neurons in these brain organoids, and what's the upper limit? Can we actually someday maybe able to culture it into becoming like a full-fledged brain of sorts? And I'm also maybe kind of throwing in one more question. When you grow these brain organoids, are these like specific brain parts, or is the brain organoid of the entire brain?
I'll answer your last question first. We grow typically fairly specific parts. So we'll target, for example, like the dorsal forebrain, right, which is predominantly cortical, or we'll target like the midbrain or the lower, like the lower brain, hindbrain. So there's a choice there with what we want to target. Capabilities, yeah. So what we've been spending the last roughly 2 years on, little 2 and a bit years on, is actually we had this nice pilot data with the Pong, and we started to explore other capabilities. This was back in 2021, long before we published. For those not familiar with the scientific peer review process, it is rigorous, but it's not quick. So we spent well over a year in the peer review process, gathering more data and doing a lot of other things. So we'd spent that time also seeing what else could we do. And what we found was that there was some promising evidence on a range of other simple tasks, but changing the software, changing the hardware to make it work was incredibly difficult because we hadn't built our own system from the ground up. A lot of the software, we'd hacked existing hardware. We were using other people's cell protocols developed. This is the way science works, right? You build upon other people's work. And as a small team, we would never have ever been able to have started from to make that that old quote to bake an apple pie from scratch. You must first invent the universe. So obviously, we worked with what we had available, what other people had discovered, and we assembled those parts in a way with a little bit of our own ideas and had a good result. The problem is that sort of platform very hard to be extensible. And so we realized very quickly what we had to do was actually build our own platform that we had complete control of. And so we set out to build something we called the CL1. And the CL1 is actually now the world's first commercial biocomputing device. It allows you to interact with neural cells in real time and be able to provide them complex information, decode that activity, to code it. It has an integrated API that makes it very, very easy to do complicated things. It can even be accessed over the cloud. So if you don't have a wet lab, you can't grow cells because that is an entire expertise on its own. How are you going to do this? Well, you couldn't, unless we cultured the cells for you and gave you access over the cloud, which is what we do. So most of the last 2 years has really been spent building that device, which is now ready. So we're pretty proud of that. Having said that, it has been ready now for about the last 6 months. So it's really the last 6 months that we've gone a lot deeper in new applications. Before that, on our research side, we've been doing things looking at really fundamental theoretical questions, and also in the drug discovery disease modeling question, which has had some amazing results, but I think is less perhaps what you're interested in right now. And so what we've been looking at in the new capabilities are things like computer vision, based tasks such as MNIST, which involves handwritten digit recognition, other things like can they respond to Morse code differently, and we're starting to build up more complex control environments as well as optimizing and improving how you can get these things. So there's a lot going on, but actually most of our big progress on that front's just been the last 6 months since we built the new tools. Maybe a bit longer than that, maybe 8, 9, 10 months, but still very early.
Right. Very interesting. So Brett, want to get into the products, but and the application. So before the products and application, these brain organoids, because these are very, very, very interesting. At this point in time, these are mini clumps. And they hold a certain number of volume of the neurons. What are the challenges? I mean, do you think which is remaining? For example, I mean, you think that eventually, I mean, we should be able to culture these, iPSC cell and grow them into like full-fledged brain someday? Is that like a possibility?
Well, it's one direction, a very challenging one. I think it is the ultimate goal of the organoid intelligence work is to end up with something that is as brain-like as possible. And the things like disease modeling, drug discovery, neurophysiology, basic science research, it's an incredibly important area. But it is just one approach, right? While there are other approaches that, as I said, leave physiology behind and say brains are all well and good, but there's plenty of people with brains around and you can pay them a bit of money and they'll come work for you and you can get access to human brains and they're pretty good. So maybe there's something we can build that does things that at least neurotypical human brains don't do very well. For example, advanced math, other things. So there's a lot of directions and we're fairly agnostic. At the end of the day, our focus is build really good tools, use them ourselves internally, but also allow others to use them. Which approach is going to be the right one in the long run, or will they just end up merging together and you'll end up with groups of human brains all networked together? There's a lot of directions it could go. How long will that take? That I don't know. Right? Could it be 5 years, 10 years, 50 years, 100 years? The only way we're going to find that out is by doing the work and seeing how long it takes and trying to get as many people involved in that work as possible. And that's really what our key focus has been.
There is an organ shortage worldwide, and with this approach, eventually, I mean, possibly if you could culture, any organs through these induced pluripotent stem cells, your lung, your brains, and so on and so forth. Well, I mean, that would be like a very, very fantastic, like a world. You mentioned about.
Although I'm not sure how many people want a brain transplant. That might be an interesting one.
Not brain transplant, maybe other organs, but because eventually if you could do that, that'd be like really, really cool because obviously we're seeing a lot of organ shortage.
Yeah, there's a lot of amazing labs across the world who are looking at this very thing. I saw recently, I think there was the trachea that was recently replaced with someone's own 3D printed cells in there. I think it was the trachea. Um, and people have been looking at this, of course, for cardiac and liver and skin. Skin's been done for a while now, but it is still phenomenal results. So there's incredibly talented scientists and bioengineers out there who are building these up. And I do believe that'll be a significant role. It's not directly what we're focused on, of course, because we're focused very much on the brain. As I said, perhaps in many ways moving away from the idea of just trying to make a human brain in a box. That's got some concerns with it that would need to be very carefully addressed.
Brett, you mentioned about the commercial launch of CL1, your product, and you also have cloud service and API. Who have been your early adopters, and what's the surprising applications that you have discovered?
So naturally, I can't talk too much about specific early adopters or their applications for confidentiality reasons. But what I will say is at a very high level, I have been surprised by the breadth of it. The areas I'll talk about now are ones that I've talked about before, and so aren't anyone's proprietary stuff. But for example, things like the stuff like basic neuroscience research, drug discovery, we expected. That's been really great. There's some really, really great works going on there. Other things like robotics or entertainment, for example, and by entertainment, I mean like people interested in making music. That's been really great. And there's some really cool work that not using our hardware, but that we were grateful to be have a very, very small role in helping with. For example, in Perth, there's an exhibit called Revivification, which is a wonderful example of exploring this technology for music, for the purpose of music. There's also people interested in using it for, gaming, doing games and a whole range of other things. So far, I would say there's no particularly, being very careful with how I word these things because I don't want to breach anyone's confidentiality. But I'll say there's been a range of really interesting approaches that people are taking. And what's great is we don't know how or what some of them are going to work, right? Like maybe some of the things people are doing right now turn out to be amazing, maybe not. But the fact is that there's a community of people that have been willing to rapidly adopt and explore this new technology. And if anything is going to make this a breakthrough success that can change the world, it's going to be this approach of having a community of people working together. So that's been really gratifying.
Right. Brett, this seems to be like a relatively new field. I mean, for the past couple of years, it's been you guys as well as Final Sparks. And I had the privilege of interviewing somebody from the CEO from Final Sparks. Who are the other peers in the field?
I saw.
Yeah. So who are the other peers in the field who are working towards organoids or bioengineered intelligence, these dual approach that you're working on? Because I believe there's some folks from China too, right, who are working on this field?
Absolutely. Yeah, there's some amazing folks out of Tianjin University, as well as some of the other universities who are doing some really, really interesting work. Of course, over at Johns Hopkins University, as I mentioned, Lena Smirnova, Thomas Hartung are leading the organoid intelligence field with a large collaboration of really talented scientists and engineers and ethicists and philosophers. That's a really great collaborative work. There's a number of, there's so many other researchers whose work, either directly or indirectly, will touch on this. We've been working closely with, for example, like Hideaki Yamamoto out of Tohoku University in Japan, who does beautiful modular work. And then there's a range of people that we don't work with, but we see their results come out and they're doing some really remarkable stuff. So I could talk all day about the different people and the different work going on, as I said, either direct or indirect. Just today, I saw a new research come out of, I think it was Harvard, looking at building a whole range of different cell types that we're keen to explore, but we didn't have the protocols. And these guys have developed a new protocol. Their main focus was on Alzheimer's and Alzheimer's research, but you could adopt it and build healthier versions of these and use them in the system. So there's a huge collaborative community going on here.
Yeah, very interesting. I've also spoken to Thomas Harting on the podcast. Are there any interesting partnerships that you have forged that you would like to share with the audience?
One of the ones though that has been public actually that I can comment on, for example, is IOHK, which is from the cryptocurrency group related to Cardano. They've been very early adopters of this technology. I can't speak to the use cases, of course, but they were kind enough to publicly sort of talk about the technology and their excitement for using it. Um, so this is very exciting to see that people from the tech space have adopted it. There's also a range, of course, on the wet lab side, the pharma and the biotech adopting it as well. So it really makes me even unsure about where some of this stuff is going. Uh, of course, that's why we're so careful in how we go forward with it. But it really makes me feel like it could be accelerated much quicker because of when you get a lot of different people with the different skills coming together, you can actually have this convergence and it can have a multiplicative effect on technology acceleration. So that's amazing to be part of.
There are a bunch of people, obviously, when any new technology happens, and specifically, when you kind of like tinkering around with what is alive. I mean, there's obviously, people get all worried, So specifically with, because right now you're saying that what you're talking about is biological living cells, these are mini brains. So there are critics who worry about, biological slavery in these wetware servers. And they how do you address this concern about these mini brain, suffering and things like that?
We actually do, at the high level, share the concern and believe it is important to explore this carefully because ultimately we need to have enough humility to acknowledge what we know we don't know, let alone what we don't know, right? Which is probably what we don't know is probably bigger, but even what we know we don't know is still pretty big. And so we've been working with ethicists and philosophers about this for a really long time. And while contextualizing the work, which is what I was referring to before, is important, and the fact is we're using tiny clusters of cells, even simpler than a bumblebee or a mosquito or a cockroach. And very few people have concerns about squashing a mosquito because it has some neural cells in there. It's still important to be aware of where the work might go. And so we've basically proposed with a range of independent ethicists and philosophers and regulatory people some actionable pathways forward. An update on this will actually come out soon from a large collaborative group from predominantly the Asia-Pacific region, but there'll be a few other authors on it as well. But what we proposed for our specific field is, one, we need to define the language because there's no point us trying to work out the ethics if we don't agree what we're talking about. And when you talk about things like consciousness, sentience, intelligence, there's no agreed definitions to the words. I remember I found a paper once that had found 52, I think it was, definitions of intelligence. A few years ago, I read it. And then by the end of it, they proposed their own, which of course meant now there's 53 published definitions. So we need to agree, like, which of these 52 definitions are we talking about if we say intelligence? Which definition of consciousness? Otherwise, you can't have ethics. Like, it sounds like kind of a weird problem to have. But when you're talking about ethics, understanding each other is such an important first step. The second step that we've also been working on, these don't have to happen exactly one at a time, although they do need to all be done eventually, is to define the metrics. So if you're saying something like consciousness is there, you should be able to have a marker of consciousness or several markers that tell you this is maybe conscious, or this has agency, or this has sentience. There should be something there to tell you it, because right now the systems almost certainly do not have it. But what about a bee? Is a bee conscious? I don't know. What do you think? Is a bee conscious? A single bee?
I believe in the panpsychism approach, where I think everything is conscious. Your single atoms to anything and everything. But like you rightfully pointed out, I mean, I don't think we have like a definition whatsoever of what intelligence is and consciousness. Obviously, consciousness goes into that extremely like woo-woo zone. I mean, we don't understand it, and here we are at the cusp of trying to build these machines which we want it to be general purpose intelligent, and I don't know the world that we are kind of getting into. I mean, it seems so exciting because even these statistical data and data out models, who would have thought, I mean, launched in 2020 three, and just it's 2022 November, and it's 2025, and what it can do is so amazing. I mean, none of there is obviously one side. I mean, none of the group of people saying they try to undermine it, but I think it's purely magical what these models are doing. I mean, it's there are these papers which are debatable, that they have emergent properties, but there was these also, like I think the Anthropic papers which was released last year, which said that these models could fake alignment, and they were showing self-preservation behaviors. So even these data and data out models, I mean, are like you said. I mean, it's we really don't.
This, right? Is this system conscious? Like I personally wouldn't say so, but if you do, then do these systems need protection? This is the complexity of what's being asked, and so actually one of the work we've done is trying to take all of this into account, and there's an ongoing call for collaboration in the scientific space at the moment, and we have about 50 different academic labs from around the world, probably more now. We're hoping to get to about 100 from all the different fields. And we're looking at trying to define a language in what's called diverse intelligence systems. And the idea is that, can we find some sort of way to talk about an LLM that is also applicable to how we might talk about the brain, or how we might talk about a slime mold, or an animal? Because if we can do that, then we suddenly can have these very interesting conversations like what you and I are having, where we can say, all right, is a bee conscious? Is an LLM conscious? Is that the same sort of consciousness? Is the consciousness of a bee so alien to you and me that we don't even consider it comparable? Or is there some similarity? Same for an LLM. Is it so alien, despite the fact that it talks back at us in whatever language we've trained it in? Is it actually so alien that we don't feel like it needs ethical protection? Which is often the way we feel about insects, that if they have a consciousness, they don't need ethical protection. So it's work that we take very seriously and is incredibly important, but it's not anything that right now needs an urgent reaction to it. Like, there's no need to ban or overregulate this work now, because especially for our work, maybe some of the LLM stuff needs more regulation given where some of it's going, especially with Sam Altman's recent announcements. It is important to be aware of the choices we're making, and choosing not to act is a choice as well, right? So if we're saying, well, we don't need regulation now, we should be able to justify that, and we do that. But we should be aware also of where those lines are, where we say, okay, now we've got to that level of complexity where there needs to be some thought and consideration. Because even if you acknowledge that everything, like your approach, I don't agree with it, but if we acknowledge and you say panpsychism is what I believe, what you believe in this case, therefore everything's conscious, well then how do you justify, say, like animal testing? If you say, well, we don't want babies to have brain damage when they're born, and there's diseases that are going around and we want to fix it. I personally believe it's necessary, but I don't, to do animal testing, but I don't believe it's the most ideal approach. And if we could avoid animal testing, that is much better. I think alternatives to animal testing are incredibly important. Animals don't have a good time in animal testing, but we believe ethically it's necessary. This becomes a question: when is it ethically necessary to use something in this way if it has consciousness, right? And obviously where that is, we draw different lines. At the moment, these neural systems we work with, almost certainly 99.99999% don't have anything that most people would call consciousness. But what if you needed a system with consciousness to cure Alzheimer's or to cure SM1, which is a horrible disease that can affect babies? Is that worth it? Most people will say yes. So we just have to draw lines. One, we need to define the language. Two, we need to figure out the metrics. Three, we need to decide what is ethically acceptable for us as a worldwide global society.
And then there's no consensus in any of those three.
There's not, but we can work towards it. And we're really trying to be part of that mission, working with a lot of independent people who, you said some people disagree with us. One of my proudest things is the fact that some of those people who used to disagree with us are now co-authors and collaborators, and we're working together to find out what we do agree on. So you can do this. Science doesn't have to be adversarial. It can be collaborative.
True, exactly. I think the world has become so adversarial. It needs to be collaborative, and yeah. So your team has authored a paper recently in August 2025 that showed that brain cells learn tasks faster than machine learning algorithms. It would be great if you could elaborate on the paper.
Yeah, we had the first evidence of that back in, I think, 2022 or 23. But we had I said at the start, peer review is rigorous, but not always quick or seldom quick. This was the slowest paper we've had. I think it was about 3 years or something of peer review nearly. But we got it published. We had enough evidence eventually to convince them. What we were testing here is something called sample efficiency, which is how much data does a system need to show learning. And machine learning, for example, can learn to play Pong, for example, incredibly well, better than you or I can, right? They'll play perfectly, but they will need tens of thousands, hundreds of thousands, millions, depending what algorithm and what the task is, of examples to do so. And then if you change anything at all, they'll break, right? But what is biology good at? It is good at getting good enough, right? When I pack away the dishes or if I make a cake, I don't do it perfectly. I do it good enough. If I drive a car, I don't drive a car perfectly, despite my best efforts, but I do it good enough. This is how biology works, and that allows us to adapt and respond in this chaotic, dynamic world we all live in. And so we found that biological neurons were also pretty quick at getting good enough. So it took them maybe, I think, less than 5 minutes of real-time data to learn. And so in that way, they learn much, much quicker than the machine learning that in real-time terms would be taking weeks or years or months, depending, again, the algorithm. Some learn quicker than others. But yeah, many, many times more data than what the biology, even a very simple biological circuit would do.
Right. Brett, today, I think the world is racing towards building what they call artificial general intelligence, general purpose machines, and there are various labs, startups, big tech companies working towards that approach. Do you see a hybrid approach where biological computing substrates are combined with these large language models, maybe possibly unraveling general purpose machines with its complementary strength and weaknesses?
Yeah, absolutely. I think, as I was saying towards the beginning of the chat, it's the right tool for the right job. So being able to figure out, where do you use a CPU? Where do you use your GPU? Where do you use your biological system? Where do you use maybe a quantum system one day if it's brought into commercial feasibility, right? There will be different pros and cons for each of these. You're neuromorphic. There's a lot of ways of processing information. The problem we have is that a lot of our discussions around them lack nuance, right? And so the idea has to be, this is going to solve every problem forever, but it won't. That's just not how tools work. A hammer is super useful, but you can't build a house with only a hammer. So integrating them is 100% going to be necessary to go beyond single purpose tasks. Always. General intelligence, or at least a superintelligence, I think, will require an integration of all the different components.
So, Brett, my last question to you. What's your personal moonshot, and where do you see Cortical Labs in the next 10 years? And it will be great if you could paint a picture of the next 10 years. What does the world look like with this adoption possibly of, biocomputers that you're building? Is it going to be utopian, dystopian, or protopian?
Yeah, sure. Like for us currently, we're really committed to getting the CL1 device out there and getting as many people to be able to access this technology as possible. Scientifically, we're trying to explore the really deep fundamental questions of how these systems work. Broadly, the way I describe it sort of in a scientific term is that any dynamic system can be understood by looking at the internal dynamics of the system and then the structure those dynamics are in. What do I mean by that? Consider a flock of birds. We would need to understand both how does the bird fly individually, and how does the flock behave in its external environment? How does it respond to an eagle, or how does it respond to food? Right? You understand the structure of the environment that the system's in and the dynamics of the system, you can understand it. For brain cells, they're slightly complicated because their structure is both informational and physical, right? For example, we talked before about organoids versus bioengineered circuits. So we want to understand and try with a range of different cell types, with a range of different structures, with a range of different information structures, and try and understand fundamentally how do these brain cells work, because we believe that's a long-term goal to further accelerating their abilities and the capabilities and the applications of this technology. In line with that, we're also doing some more immediate tasks, such as some drug discovery, disease modeling, trying to understand some basic neurobiology better. So there's a whole range of things this opens up. And what's really exciting is that despite us being a relatively small group, because the CL1 system makes it so easy to explore these questions, we can do it much, much quicker than we've ever been able to before. So that's really exciting. Talking about 10 years from now, it's always a great question. People love to ask the 10 years from now. And the truth is, we don't know for sure where it's going. There are a few areas we really hope to see evolve, one of them being personalized medicine. Because we can make brain cells from anybody who can donate a little bit of blood, it's perfectly reasonable that people with specific genetic diseases, or even with very regular diseases that aren't fully based on only genetics, would be able to give us, say, a sample of their blood. For example, my brother-in-law has both severe nonverbal autism and epilepsy. And because he can't comment on it, the only way the doctors can test whether an anti-epileptic works for him is by giving him and using him as a human guinea pig and giving him all of the different drugs one by one, slowly over time, over years and years. And then they see, okay, well, this one he has a seizure every month. This one he has a seizure every 3 months. At this dose, he has it every 6 months. Oh, but at this one, he seems to be having some cognitive impact, but we don't know because it's nonverbal. What if we could just grow some cells and set up 30 units and test for a couple of months and say, alrighty, there's 30 anti-epileptics. These 3 have the best result. And we did recently publish a paper this year showing on a test with epilepsy, for example, not unrelated to my own situation, just because it was a good model. And so this is quite compelling. So I think setting that up into a personalized medicine approach could be hugely impactful for people. But also I hope for us to be able to have far more complex control systems built out of the neurons, perhaps integrated in some early robotics, perhaps integrated in more advanced entertainment systems, gaming, and so forth. That's sort of where I see us going for the next 10 years, and a whole range of other improvements. But that's that in itself is probably an hour-long talk.
Brett, thank you so much for taking time and being part of the podcast. Yes, I think we're sitting in such a fantastic point of time today, and all of these tech stack, I think it's converging, you stem cell research, genetic editing, regenerative medicine, these organoids, bioengineered intelligence, artificial intelligence, quantum computing. And I think we just seeing the first glimpse of what the world could look like when all of these converge. Because I believe in the next few years, I think all of these tech stack is going to converge. And I think, if we take the right decision, the world is going to look so fantastic. And I really hope and I wish that we take the right decision, because I think we are in a race of sorts where the entire world is trying to acquire a certain tech to gain a certain advantage over the others. It's always been like a game of power, and I hope that it's a game of collaboration where we collaborate with each other, because there's so many problems. You spoke about your brother's issue. There's so many. I think there's 3 billion people today in the world who've got neurological disorders. And with these organoids, we do disease modeling, drug discovery. There's so many new avenues that we kind of open up. And with this convergence of all these tech stack coming together, possibly maybe there might not be any disease, And what a beautiful world that would be. More power to you guys, Cortical Labs. I think you guys are at the frontier of doing something extremely exceptional, playing around with, the wetware, maybe able to reverse engineer the human brain and possibly build general purpose machines, So kudos to you guys. Wish you guys the very, very best. And to my listeners, if you like what you see and hear, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you. Thank you, Brett. Really appreciate this. Yeah.
Thank you so much. Pleasure to chat with you.